So we kind of dealt with the internal regulators yesterday, right? How we regulate the cell cycle. The cyclins and the CDKs, right? They play the major role with internal cell cycle regulation, right? The rate of cellular division. But there's not only internal controls here, there are external mechanisms. We've mentioned HGH. We've talked about signal transduction pathways, tyrosine kinase receptors, G protein receptors. All of those can be part of regulating the cell cycle. So we'll talk a little bit about growth factors, and then we'll go into what happens when this system fails, right? When cancers form. So, gracias. All right. So there are numerous types of growth factors in our system, right? HGH, human growth factor, is growth factor. There's another category of platelet-driven, even though it's platelet-derived, it's written wrong up there. Platelet-derived growth factors, and these are growth factors that help to regulate the cell cycle. These are the signal molecules that can bind to tyrosine kinase receptors and start a signal transduction pathway, which in turn will call for transcription factors that increase the expression of cyclins. That makes sense? Right? So we have the signal molecules, right? These platelet-derived growth factors are released into your bloodstream. They are going to target tyrosine kinase receptors, causing them to form they come together and form a what? A dimer. When that dimer is formed, they phosphorylate each other and start this transduction pathway, relaying the message from the surface of the cell through the cytoplasm to the nucleus of the cell. The cellular response would be to call for specific transcription factors. Those things turn on and off expression, right? Specific transcription factors which would increase expression of cyclins. In turn, moving us through the G1 phase, right, into possibly S phase and through the rest. So that's really what external growth factors are. They're just growth factors that we release into our blood that can increase the rate of cellular division. How do they do it? They're going to increase production of specific cyclins and move us through the cell cycle. They're going to relay to the internal factors or the internal regulation system. Okay. So a couple things. It's a couple types of characteristics that normal cells display. And we're starting to get into the talk about cancer cells, about tumor cells. So a couple types of characteristics are density-dependent inhibition and anchorage-dependent inhibition. Remember, cells, we have the extracellular matrix on the outside. Cells like to be attached to other cells, especially if you are a multicellular organism, which is what we are. Right? So our cells do not do well by themselves. If you just take a skin cell and put it in a test tube by itself, unless you do some, create a proper environment, that skin cell is not going to just grow on its own. They want to be anchored to other skin cells. Another one is density-dependent inhibition. And this relates to your skin, right, is not growing, and you're not forming these large folds of skin that start kind of growing out your body and start dragging on the floor, right? Your skin knows that, hey, it only needs to grow so much. It only needs to grow enough to cover your skeleton, to cover your muscles, to keep your blood in and all that good stuff. Right? How do they know that? Well, because there is density-dependent inhibition. Right? Cells know when they've filled the area they're supposed to cell. They come in contact with one another, right? Glycolipids, glycoproteins communicate, saying we've now filled the space. We no longer need to progress quickly through the cell cycle. We need to just kind of maintain and slow down the cell cycle. So our cells typically will display that. So what we see here is we see kind of our nice little petri dish. It's just a square petri dish opposed to a round one. We have cellular growth going on the medium at the bottom. Some cells will grow well in a broth. Other cells need to be on some type of surface. So you might have some type of agar surface. Agar is kind of like a gelatin surface. We'll play with some agar later on. So here we have a nice agar surface. These cells are probably going to be undergoing cell division at a fairly high rate. They're going to be producing cyclins. They're going to be progressing through. As they start to cover the bottom, they're going to be in contact with one another. Right? They're going to start to slow down the rate of cell division. Right? Density-dependent inhibition. They're going to inhibit the cell division of the cells around them. If you were to take some cells out, the cells in this area would no longer be stopped by density-dependent inhibition. They would recognize their space to fill. So these cells in this area would most likely increase their rate of cellular division to fill that space, and then they would kind of complete filling that space. This relates to what happens if you get a cut. Right? If you cut yourself, right, you damage the cells in that area, there's now space for those cells to grow. There's clotting factors, there's a lot of other things going on, but eventually your cells are going to regrow and fill that empty space. What happens though is when you have a cancerous cell, it's no longer responding to these environmental factors. So it's no longer really responding to. Is it hot in here? It's like really hot in here. Right now. Okay. So as you can see here, right, these cells are no longer adhering to the density-dependent inhibition. They're just growing. Right? You leave this petri dish of cells, you go away for a week, you come back, they're spilling over the top of your petri dish. Not a good thing. It's not a normal thing cells would do. So these cells are growing, dividing no matter what. Nothing is slowing down their rate of cell division. So what's gone on here is most likely some type of mutation that has mutated a regulating factor within the cell cycle. All right, so genes that are involved in signal transduction pathways, genes that are involved with producing cyclin-dependent kinases or cyclins, those genes might have become mutated. And instead of being turned on and turned off and regulating the cell cycle is just constantly in the on position. So let me just kind of warn you here. This is a very simplistic view of cancer. This is kind of the real basic view of cancer. It's not that it's wrong, it's just very basic. We have a greater understanding of how every type of cancer is genetically different from other types. In fact, even if I have one type of cancer, let's say I have leukemia, leukemia in my system might not be operating the exact way leukemia in your system might be working. So this is just a very kind of general overview, a general breakdown of what happens when something becomes cancerous. But we're realizing more and more with personalized medicine that you really need to treat the individual. And not only that, even within tumors, we're realizing that cancer cells within tumors actually can be genetically different from one another. So you might need one type of treatment to treat some of the cells that might be in a cancerous tumor, and another type of treatment to treat the other cells in a cancerous tumor. So just realize we are getting better and better at treating cancers because we're getting more specialized. We're able to understand what each individual cancer is doing. So this is just a real general overview, just kind of an FYI before we go forward. So in general, there are different factors that cause cancer, right? We have genetic factors, right? And we have environmental factors, right? Environmental factors are carcinogens. So key carcinogens, obviously smoking. When you smoke, there's a lot of harmful chemicals. Benzene is one of them. Benzene is a ring structure, and it's a ring structure that's very flat. And that benzene molecule, when you take it in, can actually fit in between your DNA nucleotides. And when it fits in between your DNA nucleotides, it causes mutations. Some mutations don't matter. Some mutations do. Radiation, right? If you go out in the sun every day, UV radiation, right? That energy, that UV light can actually affect and cause your DNA to mutate. Specifically, it causes what we know as thymine dimers. I've mentioned that before. When you have two thymines that are close to one another, it'll cause them to bubble up and form a dimer together, which is mutation. And then viral infections. So different types of viruses can cause cancers as well. So we have the environmental factors. We also have genetic factors, right? We have key genes that are known as anti-oncogenes, genes that prevent cancer. We have key genes that are known as oncogenes, genes that, if mutated, can cause cancer. So we have both these going on. There's no silver bullet. There are people who have lung cancer who have never smoked a day in their life, right? And I have my uncle who smoked his entire life, has never developed lung cancer, right? So it's not a silver bullet. It's not an either or. That's why we realize your genetic makeup plays a major role with this. Some people might have a very, very hefty supply of anti-oncogenes. These are genes that if the cell were to become cancerous, these genes destroy those cells. So maybe if you have a hefty dose of anti-oncogenes that are working very well, smoking never really caused cancer in your body. Yes? Well, that's one of the major, I'll talk about one in particular, and that's a key focus of some type of anti-cancer drugs. There's a lot of different ways they go about it. Okay. So we have anti-oncogenes, we have oncogenes. So just realize this is very specialized, it's very different. So let's talk about anti-oncogenes first. And the key one, which we should know about, and this will very well be on the AP exam, definitely will be on the SAT2 exam, it always shows up, is P53. P53 is a key anti-oncogene. What part of the cell cycle would P53 show up in? Where do you think P53 would be the most active within the cell cycle? Where are we going to recognize mutations? And if we recognize mutations, we are going to destroy the cell. So you got it. G2 phase, right? G2 phase is the checkup phase. That's where P53 is the most active. So P53 is an anti-oncogene. You want a healthy P53 because we all have cancerous cells in our body. We all have cancerous cells right now, right? What happens, P53 is not the only one, but it's one of the major role players in this. P53 is activated when there's a mutation and a cell looks like it could become cancerous, could become a tumor. What happens is P53 is activated and P53 starts off the apoptosis pathway. Right? It starts to a programmed cell death and it destroys the cell. People have different forms of P53. You might have the healthy form, right? The weakened form, or the ineffective form. In fact, if you kind of saw the stat before, I got that stat a while ago. I don't know if it still holds true at 50%, but it's a very, very high number of all cancers can be related to having a weakened P53. So I don't know what the percentage is. Just realize that having a faulty P53 makes you susceptible to cancer formation. So there's a large percentage of people with all types of cancers that might have a faulty or not 100% optimal P53 gene. So this is known as just like an anti-oncogene, right? If it's working well, it's preventing your cells from becoming cancerous. This right here is we're really just kind of looking at a tyrosine protein kinase. I'm actually going to pull up a different image just to make it a little bit easier to deal with it. Not that this image is horrible. It's fine. Let me just pull up a different one, though. Give me a simple view. There we go. Okay, so here's kind of a more simplistic view of what we're just looking at. It's really looking at kinase proteins. So this is our familiar tyrosine kinase receptor, and the genes associated with our tyrosine kinase receptor could be equivalent to oncogenes. They're not genes that necessarily are bad, but they are genes that express proteins that are major players in the regulatory process of the cell cycle. So the genes that form the tyrosine kinase would be considered oncogenes. If I were to have a mutation in my tyrosine kinase receptor, and this receptor perhaps forms a dimer that is unable to separate, therefore continuously phosphorylating, that could be kind of the starting point of a cancerous growth. Right? So that would be a great example of an oncogene. Same thing could be with a G protein, or any of the proteins that are involved in the cascade, in the transduction process. Any of those genes could be considered, or those proteins could be considered related to oncogenes as well. So it's just kind of the loss of the regulatory factor of the pathway. So just to give you an idea what this is talking about, this is just kind of going between the inactive form and the active form. right? When you phosphorylate it, you're activating it. When you dephosphorylate, you're not activating it. If I remember correctly, I believe the mutation is showing, if you see the longer tail here, what they're trying to show here, and you see the shorter tail here, that shorter tail is kind of relating to the mutation that happens. Therefore, this can't be dephosphorylated. That's why it's showing it. But it's easier to understand on the tyrosine kinase receptor, what we saw before. All right. So, one kind of last thing we'll talk about. We'll leave that for tomorrow, and we'll do this. Okay, so that's it. That's really the kind of basics on cancer. It's just simply looking at oncogenes versus anti-oncogenes, and just kind of understanding that a breakdown in the regulation of the cell cycle can lead to cancerous growth. However, it is far more complex than that these days. So what I just said is not wrong. A breakdown of the cell cycle is what causes cancers. But what we're starting to understand are that there are a lot of complex factors that can lead to the breakdown of the cell cycle. Dr. Bissell, I almost forgot her name, but Dr. Bissell was looking at and starting to understand that we think of cancerous growth as being an internal cell process, a problem with the internal regulatory system of the cell. And what she's come to understand in some of her studies of some of her cancers, it actually might deal with a little bit of the microenvironment that surrounds the cell. So not necessarily the cell itself has had issues, but the environment that cell is in is causing that cell to become cancerous. And that's a very, very different way to look at how we might treat cancers, because it's getting a little bit away from the genetic pathway breakdown to more an environmental breakdown. Before we do though, what are some typical ways? What are some typical ways that we treat people with cancer? So some of the kind of standard ways that we treat people with cancers. Yes. Right. We use chemotherapy. Chemotherapy are drugs that try to target fast-growing cells. They try to target fast-growing cells, therefore they're targeting cancer cells. When you have chemotherapy, what oftentimes happens? Right, you lose your hair, right? You lose your hair because your hair follicles are fast-growing cells. That's why they're affected. Oftentimes you can get sores in your mouth as well because your skin cells in your mouth are fast-growing as well. So chemotherapy is one major way that we treat it. They're getting better and better at targeting. It's not like there's just one type. There's many, many different types of chemo drugs. There's different cocktails. There's different things to target different types of cancer. So chemo's one. What's another one? Right, radiation. So using a more focused radiation therapy, where if you actually have a tumor in a lymph node possibly, they might be able to use a focused radiation to try to target that specific area. What else? Surgery, yeah. So I mean, one of the key things they'll do is they usually have a combination. If you have a localized tumor, you're somewhat lucky in a sense because it's localized. So they might try to give you a little bit of radiation, a little bit of chemo to try to minimize the tumor, and then they might go in and actually extract it. They might do surgery and actually cut it out. That only works though if the cancer hasn't done what? What's the big word in cancer it's kind of the word that people don't want to hear? Spread. What's the word for spread? Starts with an M. M-E-T. Bless you. Metastasis, right? So metastasis is what we don't want to hear. So metastasis is what happens when the tumor goes from being localized to the tumor actually spreading. And that's a point where it becomes far more difficult to treat. You can't localize your treatments. You can't extract the tumor. Now that those cancer cells are spread around your body, and it becomes far more difficult to get all of those cancer cells. You might go into remission, but it's hard to really get rid of all of them. They might be there for a period of time. So metastasis is really what we try to avoid, but it definitely happens. There are a number of different ways that we're kind of looking at treating cancers. James definitely mentioned, how could we possibly use that p53 pathway? And that's definitely a focus of a lot of research, is trying to identify drugs that could activate the p53 pathway. Another key one, which is kind of like an up-and-coming one, is immunotherapy. It's actually trying to get. What's the problem with this? The problem is we're trying to kill cancer cells. Well, it's easy. Why don't we just go in and kill the cancer cells? The problem is your cancer cells are your cells. So how do you differentiate between the cancerous cells and between your cells? What they're starting to realize with certain types of cancers, there actually are specific receptors that do differentiate them. So one of the new things they're doing is they're actually looking to use your own immune system against cancer cells. They might take out some of your white blood cells, right, and they might they might genetically alter them to target specific cancer cells in your body. So it's kind of an immunotherapy type process. It's not extremely common, but it's something that's kind of up and coming that seems to be kind of successful. So there's a lot of different ways that we can kind of treat it these days. We're definitely better at it. Yes.
It's like diet or something to treat cancer.
I don't know about treating cancer, but I would definitely say about preventing cancer. Realizing more and more is the environmental factors, what you do to your body on a daily basis, could definitely lead to cancer formation. So simple things as obviously not smoking, not drinking, things like that, but eating a healthy diet, staying hydrated. Staying hydrated could be a major player in cancer formation, right? Just because you're keeping your body hydrated, your cells are flushed, right? Your digestive system is cleaned out a little bit more. Eating healthy foods is definitely going to play a role as well. So not necessarily, I don't know so much about treatment. I mean, if you're going through treatment, it's definitely not going to hurt you. It's only going to help you be strong and fight through it. But definitely for prevention, I'd say absolutely.
Can we insert the unhealthy p53 gene via virus?
We're getting there. There's the problem is you need to target all the cells in the body. If they have a faulty p53, that means all of your cells, the trillions of your cells, have a faulty p53 gene. Now, if you have lung cancer, I don't know, maybe that is a possibility. You could use kind of an influenza-type viral vector to put the correct p53 in. So possibly, it's definitely a target. The other one is there's a new system out there known as kind of like the CRISPR system, the CRISPR-Cas9 system, which is kind of giving us the ability to target specific genes in an organism. For the most part, we were genetically modifying something. We could do it in vitro very easy. We could take a cancerous cell, knock out the p53, put in a good p53, no problem. But the problem is doing it in a whole organism. How do you target that specific location? There's actually new things coming out, new mechanisms that we actually have the ability to do that. The mechanism is still kind of going through, it's still being vetted, it's still being tested. So it's not something we're using in humans yet, but it's definitely something that could be there. Yeah. All right, with that said, let's watch this. At least get through some of it. She has a little bit of an accent. Have you seen this one?
Now, I don't usually like cartoons. I don't think many of them are funny. I find them weird. But I love this cartoon from the New Yorker. So the guy is telling the cat, don't you dare think outside the box. Well, I'm afraid I used to be the cat. I always wanted to be outside the box, and it's partly because I came to this field from a different background, a chemist and a bacterial geneticist. So what people were saying to me about the cause of cancer, sources of cancer, or for that matter, why you are who you are, didn't make sense. So let me quickly try and tell you why I thought that and how I went about it. So to begin with, However, I have to give you a very, very quick lesson in developmental biology with apologies to those of you who know some biology. So when your mom and dad met, there is a fertilized egg, that round thing with that little blip. It grows, and then it grows, and then it makes this handsome man. So this guy, with all the cells in his body, all have the same genetic information. So how did his nose become his nose? His elbow, and why doesn't he get up one morning and have his nose turned into his foot? It could. It has the genetic information. You all remember, Dolly, it came from a single mammary cell. So why doesn't it do it? So have a guess of how many cells he has in his body. Somewhere between 10 trillion to 70 trillion cells in his body. Trillion. Now, how did these cells, all with the same genetic material, made all those tissues? And so the question I raised before becomes even more interesting if you thought about the enormity of this in every one of your bodies. Now, the dominant cancer theory would say that there is a single oncogene in a single cancer cell, and it would make you a cancer victim. Well, this did not make sense to me. Do you even know how a trillion looks? Now let's look at it. There it comes, these zeros after zeros after zeros. Now, if 0.0001 of these cells got mutated and 0.00001 got cancer, you'll be a lump of cancer. You'll have cancer all over you, and you're not. Why not? So I decided over the years, because of a series of experiments, that this is because of context and architecture. And let me quickly tell you some crucial experiment that was able to actually show this. To begin with, I came to work with this virus that causes that ugly tumor in the chicken. Rats discovered this in 1911. It was the first cancer virus discovered, and when I call it oncogene, meaning cancer gene. So he made a filtrate. He took this filter, which was the liquid after he passed the tumor through a filter, and he injected it to another chicken, and he got another tumor. So scientists were very excited, and they said, this single oncogene can do it. All you need is a single oncogene. So they put the cells in culture, chicken cells, dump the virus on it, and it would pile up, and they would say, this is malignant and this is normal. And again, this didn't make sense to me. So for various reasons, we took this oncogene, attached it to a blue marker, and we injected it into the embryos. Now look at that. There is that beautiful feather in the embryo. Every one of those blue cells are a cancer gene inside a cancer cell, and they're part of the feather. So when we dissociated the feather and put it in a dish, we got mass of blue cells. So in the chicken, you get a tumor. In the embryo, you don't. You dissociate, you put it in a dish, you get another tumor. What does that mean? That means that microenvironment and the context which surround those cells actually are telling the cancer gene and the cancer cell what to do. Now, let's take a normal example. The normal example, let's take the human mammary gland. I work on breast cancer. So here is a lovely human breast, and many of you know how it looks, except that inside that breast, there are all these pretty developing tree-like structures. So we decided that what we like to do is to take just a bit of that mammary gland, which is called an acinus, where there are all these little things inside the breast where the milk goes, and the end of the nipple comes through that little tube when the baby sucks. And we said, wonderful, look at this pretty structure. We want to make this a structure and ask the question, how do the cells do that? So we took the red cells. You see the red cells are surrounded by blue, other cells that squeeze them, and behind it is material that people thought was mainly inert, and it was just having a structure to keep the shape. And so we first photographed it with the electron microscope years and years ago, and you see the cell is actually quite pretty. It has a bottom, it has a top, it is secreting gobs and gobs of milk because it just came from an early pregnant mouse. You take these cells, you put them in a dish, and within 3 days they look like that. They completely forget. So you take them out, you put them in a dish, they don't make milk, they completely forget. For example, here is a lovely yellow droplet of milk. On the left, there is nothing on the right. Look at the nuclei. The nuclei on the cell on the left is in the animal. The one on the right is on a dish. They're completely different from each other. So what does this tell you? This tells you that here also context overrides. In different contexts, cells do different things. But how does context signal? So Einstein said that for an idea that does not first seem insane, there is no hope. So you can imagine the amount of skepticism I received. Couldn't get money, couldn't do a whole lot of other things, but I'm so glad it all worked out. So we made a section of the mammary gland of the mouse, and all those lovely acini are there. Every one of those with the red around them are an acinus. And we said, okay, we are going to try and make this. And I said, maybe that red stuff around the acinus that people think is just a structural scaffold, maybe it has information. Maybe it tells the cells what to do. Maybe it tells the nucleus what to do. So I said extracellular matrix, which is this stuff called ECM, signals and actually tells the cells what to do. So we decided to make things that would look like that. We found some gooey material that had the right extracellular matrix in it. We put the cells in it, and lo and behold, in about 4 days, they got reorganized. And on the right is what we can make in culture. On the left is what's inside the animal. We call it in vivo. And the one in culture was full of milk. The lovely red there is full of milk. So we got milk for the American audience. All right. And here is this beautiful human cell. And you can imagine that here also the context goes. So what do we do now? I made a radical hypothesis. I said if it's true that architecture is dominant, architecture restored to a cancer cell should make the cancer cell think is normal. Could this be done? So we tried it. In order to do that, however, we needed to have a method of distinguishing normal from malignant. And on the left is the single normal cell, human breast, put in three-dimensional gooey gel that has extracellular matrix. It makes all these beautiful structure. On the right, you see it looks very ugly. The cells continue to grow. The normal ones stop. And you see here in higher magnification, the normal acinus and the ugly tumor. So we said, what is on the surface of these ugly tumors? Could we calm them down? They were signaling like crazy, and they have pathways all messed up, and make them to the level of the normal. Well, it was wonderful. Boggles my mind. This is what we got. We can revert the malignant phenotype. And in order to show you that the malignant phenotype, I didn't just choose one. Here are little movies, sort of fuzzy, but you see that on the left are the malignant cells. All of them are malignant. We add one single inhibitor at the beginning, and look what happens. They all look like that. We inject them into the mouse, the ones on the right, and none of them would make tumors. We inject the other ones in the mouse, 100% tumors. So it's a new way of thinking about cancer. It's a hopeful way of thinking about cancer. We should be able to be dealing with these things at this level. And these conclusions say that growth and malignant behavior is regulated at the level of tissue organization, and that the tissue organization is dependent on the extracellular matrix and the microenvironment. All right, does form and function interact dynamically and reciprocally? And here is another 5 seconds of repose is my mantra. Form and function. And of course, we now ask, where do we go now? We like to take these kind of thinking into the clinic. But before we do that, I like you to think that at any given time when you're sitting there, in your 70 trillion cells, the extracellular matrix signaling to your nucleus, the nucleus is signaling to your extracellular matrix, and that is how your balance is kept and restored. We have made a lot of discoveries. We have shown that extracellular matrix talks to chromatin. We have shown that there are little pieces of DNA on the specific genes of the mammary gland that actually respond to extracellular matrix. It has taken many years, but it has been very rewarding. And before I get to the next slide, I have to tell you that there are so many additional discoveries to be made. There is so much mystery we don't know. And I always say to the student and postdocs I lecture to, don't be arrogant, because arrogance kills curiosity and passion. You need to always think what else is need to be discovered, and maybe my discovery needs to be added to, or maybe it needs to be changed. So we have now made an amazing discovery. A postdoc in the lab, who is a physicist, asked me, what do the cells do when you put them in? What do they do in the beginning when they do? I said, I don't know. We couldn't look at them. We didn't have high images in the old days. So she, being an imager and a physicist, did this incredible thing. This is a single human breast cell in a 3 dimension. Look at it. It's constantly doing this, has a coherent movement. You put the cancer cells there, and they do go all over. They do this, they don't do this. And when we revert the cancer cell, it again does this. Absolutely boggles my mind. So the cell acts like an embryo. What an exciting thing. So I like to finish with a poem, but I used to love English literature, and I debated in college which one should I do, and unfortunately or fortunately chemistry won. But here's a poem from Yeats. I'll just read you the last 2 lines. It's called Among the School Children. O body swayed to music, O brightening glance, how do we tell the dancer from the dance? And here is Mirs Cunningham. I was fortunate to dance with him when I was younger. And here he is a dancer, and while he's dancing, he's both the dancer and the dance. The minute he stops, we have neither. So it's like form and function. Now, I like to show you a current picture of my group. I have been fortunate to have had these magnificent students and postdocs who have taught me so much, and I have had many of these groups come and go. They are the future, and I try to make them not be afraid of being the cat and being told, don't think outside the box. And I'd like to leave you with this thought. On the left is water coming through the shore, taken from a NASA satellite. On the right, there is a coral. Now, if you take the mammary gland and spread it and take the fat away on a dish, it looks like that. Do they look the same? Do they have the same patterns? Why is it that nature keeps doing that over and over again? And I'd like to submit to you that we have sequenced the human genome. We know everything about the sequence of the gene, the language of the gene, the alphabet of the gene, but we know nothing about the language and alphabet of form. So it's a wonderful new horizon. It's a wonderful thing to discover for the young and the passionate old, and that's me. So go to it.